Thanks, Maya.
Lovely. Thank you so much for that introduction, Laura. So hello, everyone, and thank you for being here with me as I spend the next couple of minutes telling you all about the pelvis. So like she said, my name is Mayowa Adegboyega. I'm a postdoctoral researcher in the evolutionary anthropology department at Duke University. And I am actually going to be presenting you part of the work that I did during my dissertation at UC Davis. So the title of my talk is Predicting the Position of Hip Bones Within the Pelvic Girdle, and I'm using the Kabara to Neanderthal as my case study. So for this talk, I'm going to sort of go over, the function and the evolution of the human pelvis. I'm sure not all of you have spent the last couple of years obsessing over it, so I'll just do a brief overview of that. Then I'll talk about what really sparked this research project. So what's the premise and what were my overall goals? Then I'll sort of talk a little bit about my previous work with Kubara 2 and how that led to this particular project, go over the details of this study, our results, our findings, and where we see this going from here. So, as Laura just said, the pelvis is a very important part of our anatomy. It is important for a lot of human biological processes that have been critical for our survival and our reproduction. For example, the pelvis, the angle that the pelvis tilts influences our posture. The sort of hip joint connects to our lower limbs and to the rest of our bodies, which transfers load from our upper bodies to our lower bodies as we walk. The width of the pelvis affects sort of the dimensions of our body. It's sort of one of the widest parts of our body. And that because it affects the dimensions, it impacts things like thermoregulation. And of course, the one that everyone sort of thinks about immediately, it influences the mechanisms of childbirth. So the shape of your pelvis, the size, will influence how childbirth works. So the pelvis has undergone many changes throughout human evolution in response to different evolutionary pressures like childbirth, like I mentioned, or locomotion, walking, and other things. It's also undergone some changes due to neutral evolutionary processes like genetic drift or migration, and that leads to some of that geographic variation we sometimes see in the pelvis. So on the screen is just a few examples of some pelvic shapes that have occurred within the hominin lineage. And it just gives you an idea of the sort of variation that we sometimes see. So some pelvises are much wider and some are much taller. Some have iliac blades, so the upper part of the pelvis, that are much more flared than others. And these impact all those biological processes. And so looking at the pelvis can really give us a lot of insight into the life histories and the life ways of a lot of our early hominins, a lot of fossil hominins. But alas, right? The pelvis can tell us all these wonderful things if we could find them. As some of us here know, the fossil pelvises are quite prone to heavy damage and distortion in the fossil record. And why is that? Well, for one thing, right, the pelvis is this curved bone with a hollow cavity. And so you can imagine that when it's buried in the ground, I mean, the easiest thing that happens is after that pressure of the sediment above, it crushes it or distorts it. And because of that, it makes it very difficult to observe the morphology of the individual that we're looking at, or even to say anything about the biology or the biological processes that the pelvis might impact. And so because of that, when I sort of started my dissertation work, I really wanted to contribute to the reconstruction of fossil hominin pelvises to see how we can extract as much information from the materials that we have. And so I was going to use 3D geometric morphometrics to sort of develop methods for predicting missing pelvic shapes, which is what I'm going to talk to you about today. And so the process that I use is this regression-based predictive modeling that helps us to predict the position and the orientation of hip bones within the pelvic girdle. But before we get there, let's see how where this idea came from. So my first sort of dissertation work was to reconstruct the Kabara to Neanderthal pelvis. That's the wonderful thing you see twirling on your screen right now. And why did I pick Kabartu? What's so special about this individual? The Kabartu Neanderthal is essentially a male Neanderthal partial skeleton found in the Kabara cave in Mount Carmel in Israel. It's about 60,000 years old, and it's sort of a pretty important fossil because it's relatively complete, as at least compared to other hominids and other individuals. It has a relatively also pretty complete-ish pelvis. As you can see down here from a pointer, we are missing a lot of that left hip bone, but the right hip bone is mostly complete. You can't really see it in this image, but a lot of those fragments are sort of distorted and sort of not in anatomical position. So part of the work was to sort of create some reconstruction to correct some of those distortions. And it makes sense, right? Because Kabara, because it's so complete, because it's a Neanderthal, it's been used in a lot of studies to represent Neanderthal body breadth, pelvic canal shape. And so because of that, it's been used to calculate things like climactic adaptations, infant head size, birth mechanisms, and so on. So it's really important that we're dealing with Kabara as true to life as we can possibly sort of get it to. So long story short, lots of years of, work and heartbreak and all the other wonderful things that come with doing research. These research was published in Journal of Human Evolution in 2020. And so what I have here is actually two reconstructions of Kabara, and I'll explain the third. But essentially for this project, I created my created reconstruction protocols, and then I had a colleague and friend of mine, Peter Stamos, do follow those protocols to create another reconstruction. And this was to sort of observe inter-observer error, right? To see where my biases or someone else's biases were. And then we compared that to an original reconstruction done by Yolrak in 1987. Now, the thing about Yolrak's Kabara reconstruction is that it is a hemipelvis. So it's only half of a pelvis that was reconstructed because, as you remember, the left side was very, very broken. And with the technology of 1987, that was sort of the best work that you could really get done. So what we actually did was to create a virtual other side by reflecting the hemipelvis. And so that's why we have a full pelvic inlet over here, right? So the original was just sort of reflected along the midline. And then we took some linear measurements and compared the Yolrak pelvic shape to mine and Peter's reconstruction, so reconstruction 1 and 2. And one of the things that we noticed is that, surprise, surprise, both new reconstructions did not come out the same. And there were definitely some areas where choices were made that had to do with, the fragmentary nature of this individual, not knowing exactly where the bones would go, how to orient them, how to angle them, particularly because the pubic symphysis joint was damaged. So the joint right here at the front is damaged. And so it makes it easy to angle those bones in sort of almost, it gives a wide range of how you could angle those bones, leading to a lot of uncertainty in that process. So it was sort of my second goal to see how do we reduce that uncertainty and that sort of individual sort of bias in the reconstruction process. Of course, we can't do that entirely, but in any area in which we could sort of remove sort of individual decision making, I wanted to see if that was something that we could sort of achieve. We could do a quantitative reconstruction as we see here. So that's what led me to the particular thing that I want to focus on for the rest of my time. So that's how this idea came out. We wanted to do a more quantitatively determined orientation of the hip bones within the pelvic girdle. So what you're seeing here is a wireframe of like a left hip bone. Just for the uninitiated, you have the acetabulum over here, and we have, and I'll get into that later. So the reason particularly I was confident about using Kabara as my case study here is that, like I said, it's a relatively complete pelvis compared to, other fossils. And so it's a really good one to use to test a new method because while there's uncertainty, it's not as broad as uncertainty as you find with something like Taboon or other sort of hominins. So just to sort of make what I'm thinking about sort of visualize that more clearly. Let's say in the fossil record, a lot of times you preserve, let's say, one bone, maybe one hip bone. You're probably missing the sacrum, or maybe it is not as complete enough to reliably articulate the other bones. How would you essentially sort of virtually reconstruct the other side? Now, the beauty of the pelvis is that our body's bilateral symmetry allows us to essentially reflect one side to the other when we're doing a reconstruction. But where would you place the other side? Would you place it like this? Or would you place it like this? As you can see, especially when you don't have the other elements, you don't have soft tissue, you don't have a good sacrum, it's easy to sort of orient those bones in a variety of ways. And so what I wanted to see was, how do we make the best choice in how we orient those bones during virtual reconstruction? So I did this by training a reduced rank regression model, which is a sort of multivariate linear analysis, a sort of a regression analysis on a human data set, on a modern human data set. And I use that to predict what actions, so the transformation matrix, as it were, the actions it would take to make the present side, whatever side we select as the present side, into the missing side. So the object of this was to essentially figure out what instructions would you sort of give to make that side fit onto the mirror image more perfectly. And then we tested this on microbar 2 reconstructions. So to do this study, I collected CT scans of about 103 adults, so 52 biological males, 51 biological females, ranging between the ages of 20 to 96. And I collected this data from the UC Health System, so the University of California Health System. We didn't really control for population diversity, but according to sort of the US Census Bureau, California has a pretty diverse population. So I was pretty confident that the individuals in our sample will reflect a broad range of population histories. And so it sort of allows us to account for a significant portion of global pelvic shape variation. I segmented the pelvis out and then collected landmarks and semi-landmarks along the pubic symphysis and along the inlet rim. So this is just a wireframe of the landmarks and semi-landmarks collected. I did a lot of this work in Aviso and the quantitative analysis in R. So to begin our analysis, first we sort of Procrustes registered all our data set, essentially fit them to one another through a process of scaling, translation, and rotation. And to ensure that the data that we were essentially using only data from the side that is present, because essentially we're trying to model a case where in the fossil record, you only have one side. So I only Procrustes fit my data using the data of the side that we were assigning as present. And so that's what we used to Procrustes fit the data. So for this, it was the right side, the right hip bone was used to fit the data. Then we collected information about centroid size because that was important for the next step of our data, of our analysis. So to prepare our sort of predictor coordinates, because we're essentially trying to create a statistical predictive model. So to prepare that, first I had to scale up the Procrustes registered coordinates. And why do we scale this back up? Essentially, when you've Procrustes registered your data set, you've essentially scaled them to each other. You've translated them right on top of each other, and you've rotated them to fit. But that means that when I'm trying to determine the instructions to give essentially my data set to flip one side to the other side, essentially to create the mirror image of the other side, I actually need to know the actual distance between the two bones, not the scale distance. So essentially we sort of rescaled our data back up. Then I removed the sacrum because, again, we're trying to imagine a world where there's no sacrum, which is a very actually common world to imagine. So essentially you have two hip bones now from our human data set. So we have two hip bones, and essentially we only keep the landmarks from the left and right hip bone, but none from the sacrum. Then we assigned the right side as the present side or the predictor, and the left side as the target or the missing side, so to speak, to predict. So then first thing that we will do, right, is to first reflect the predictor side. Essentially, you're trying to create that target bone. So the first obvious thing to do is to reflect it, to create that mirror image to match up to the other side. Then the next thing you need to do is to translate it. So to move it over to where the other side is. And then the last thing you need to do is to rotate it properly onto the other side, right? And because while we are symmetric, we're not perfectly symmetric, some adjustments still have to be made, hence the rotation step, to make this sort of new left side sit perfectly onto the other side. So how does this then work out? Essentially, so in practice, that reflection essentially means multiplying your coordinate data by a reflection matrix that sort of mirror images the object along the YZ plane, and then multiplying that coordinate data by a translation matrix, essentially the coordinates that it would take to move them over to the other bone, and then figuring out the rotation matrix that would essentially sit that bone perfectly as you can onto the actual left side. So what we just did in R was to essentially determine what is the reflection matrix that you need to do this, what is the translation matrix that you would need to do this, and what is the rotation matrix that you would need to do this. And once we did that, we combined them into one transformation matrix. And essentially that matrix is the data set. So sorry, essentially that matrix for each individual is the instructions that it would take to create a new left side from the right side. So each individual gets its own transformation matrix because of course, each individual is different. So what we then do here is once we have that translation matrix, right? Or once we have that matrix that essentially tells each individual that if I multiply your coordinate data from your right hip bone by this translation matrix, it will produce the left side. Then we sort of fit this reduced rank regression model to that data. And we use sort of leave one out cross-validation to assess our prediction errors. Now, why do we do that? So leave one out cross-validation is actually very great for assessing this kind of model prediction, because what you do is, so we have 103 adult individuals. You essentially take the individual that you want to predict out of the data set, you train your model on the other data sets, and then you tell the model to predict for that particular individual. But because you took them out of the data set, they don't influence the results, because if you have the individual in the data set that you want to predict, you're kind of sort of biasing yourself already. You're sort of introducing the known quantity that you're trying to estimate. So we remove that known quantity out. We use the other individuals in the data set to predict this individual, and then we see how well the model performs. And so we did this for each and every individual in our sample, training our model so that our model sort of learns to sort of better make these predictions. And so then I wanted to see how well did my model perform, right? I've trained it on my data set, my human data set. And in my human data set, because I know exactly where that missing bone is, where that target is, I can compare the actual side with the prediction from my model and see if my model even sort of, does a good job. And so what I did was I essentially just took two individuals out of my sample, which is what you're looking at here. So you're looking at the left hip bone of two individuals in our sample. This one, A, represents an individual with average prediction error. So essentially, what we're looking at in A is someone whose prediction error was essentially close to the mean. And as you can see, the model does a pretty good job of estimating where that other bone actually sits. So it's very color coordinated here, but essentially the dark black lines are the outlines of the actual hip bone, and the gray lines that you're seeing are the outlines of the prediction. So same thing for the blue and green. So the blue and green lines that mark the pelvic inlet are also blue for the original, green for the prediction, and for the pubic symphysis, yellow for the original, red for the prediction. And as you can see with A, for the average individual in our sample, the model does a pretty good job of predicting exactly where that bone sits. Then I wanted to see what is the worst that this model can essentially provide. Like, what is sort of one of the worst predictions that it makes? And that's the example we have in for B. So essentially, an individual in the fourth quartile of prediction error, like sort of like the fourth quartile sort of worst predictions. And even for that individual, while we can see that the gray and the black lines are not particularly as well situated as in A, it still did a pretty good job of, predicting where it should sit. It's sort of still within what I would call a decent margin of error. So most of this error, you can see, it looks like the actual bone is a little bit more inferiorly rotated upward than the prediction, right? Which seems to be a little bit more inferiorly rotated sort of forward. But essentially, as far as like when we think about the purpose of the study for fossil reconstruction, this level of error is pretty decent. So I wanted to actually quantify, like, what does that error mean? So I created compared my sort of model sort of performance to two sort of null models to sort of see how well it did. And so over here, the yellow lines essentially represent the prediction error in my sample. So you see, most individuals have pretty low prediction error that sort of falls along a very sort of a low mean here. But then I also compared it to a mean shape model. So the mean shape model essentially is like, what would be if I compare, what would be the distance between every sample in sort of all 103 individuals to the mean of shape of that sample. So I essentially just compared the error that would occur if I compared every individual just to the mean. So if I said I was just going to use a mean shape null model, how well would the model perform? And the mean shape null model obviously performs worse. And then I also compared it to a pairwise distance model. So essentially comparing the difference between every pair of two individuals in our sample. So I paired each individual with every other individual in our sample and looked at the difference between them. And that's the green line you see on the bar. So it looks like our model is doing very much better than these two sort of null models. But doing this for a modern human where you already know what the shape is or you already know where the real bone is not the end goal, right? The end goal is to see how well this now works for a fossil hominin. So essentially, I did the same thing with my two Kabara reconstructions. So the one created by me and the one created by my colleague, Peter Stamos. So essentially what I did was essentially introduce Kabara's right hip bone into my model. So introduce coordinate data from just the right hip bone of Kabara and to see where the model will place the left side. So for the first reconstruction, that's what you're looking at here. The wireframe shows that it does a pretty decent job of estimating where Kabara 2 sits in my reconstruction. And to compare it to sort of like the human sample, we have this purple arrow down here that essentially shows the error, the Euclidean error for Kabara. So as you can see, it sits right there with, kind of close to the mean of the other prediction error. So essentially, the model is doing a very, very good job of predicting Kabara's hip bone position. Then I compared it to the second one, and again, still close to the mean, slightly offset, so a higher prediction error for the second one. And I essentially interpret this as my reconstruction is better. Essentially, but still both both reconstructions are pretty decently predicted by the model. So essentially, what's the purpose of all of this, right? What essentially I want to sort of do, hopefully I can get that done somewhere in between my postdoc research work, is to properly apply this model to the Kabara pelvic reconstructions and to actually recreate the sort of 3D reconstruction that I did according to sort of using the information that I got from this project. And then compare some of the linear measurements and geometric morphometric analysis to sort of compare that to my original reconstruction. And the idea is that we want to sort of improve the reconstruction as best as we can. Another thing that I want to do is actually improve the predictive capabilities of this model, right, for other fossil hominins. Now, Neanderthals are very similar to modern humans in many ways. And that also means anatomically, they're at least relatively more similar to us than other fossil hominins. So what I want to do is actually introduce other data into that model to help it we can sort of apply it to sort of earlier hominin species that have much more different shapes and anatomy than like Neanderthals are to humans. So, for example, I would like to introduce like chimp, data, pelvic shape data into the model and see if that will sort of make it perform better for something like an Australopith or something like the more earlier hominins. Because if we sort of apply this model now, it's too close to humans and to Neanderthals to work very well for earlier hominin species. And the idea of all of this is not to say that you don't sort of need people to do virtual reconstructions with other means. Of course, all of these things can work together. The idea is to create this method that complements other techniques that can help us sort of make better, more informed, less biased, more quantitatively driven fossil reconstructions. And if we have that, and if we can essentially do our best job of recreating these hominins as they would have been in life, we have a better chance of really making better interpretations about their biology, their life histories, and the sort of the process that led to human evolution. Thank you very much. First, I'd like to just thank the individuals whose data we used for my sample. These are real people living in California, and do not take for granted that their data is being used in research, and of course, other individuals that I collaborated with. Thank you very much. Questions or anything?
Thanks, Maya. Thanks very much. That was great. Does anyone have a question that they'd like to start with? Perhaps I'll get us started and then other people will join in. I was wondering if you just sort of looked at the effect of sex or age on the model, and then perhaps would you be able to use your model to sort of work out what the difference between male and female are in the recent human or the current human sample, and then apply that to try and create a virtual female Neanderthal pelvis?
So that's actually that's a very good question, because that's one of the things that I sort of bookmarked as like one of those later projects. The part of what I actually wanted to do originally and at Davis was to do that, create that female Neanderthal pelvis. Never quite got to it because everything gets in the way. But I didn't account for sex in this particular sample. I tried to sort of keep it balanced because I just essentially wanted to just test the model process at all. But it'd be really interesting to actually look at that, to sort of essentially introduce, like, make sex an actual predictor in the model prediction. I am a little, well, these are questions to be tested, but it is interesting because one of the sort of different things about Neanderthals is that they're in many ways, in many aspects of their pelvis, sort of fall close to the sort of female modern human shape. So even the male Neanderthals, for example, have like that really long pubic bone that creates that wider inlet. And so it would actually be interesting to see if for example, maybe you make a better prediction for Neanderthals with just using a female sample, for example, or if, for example, or who knows, maybe you actually do still make a pretty good prediction with just the male sample. So there are many things that I want to test to see what other predictors are in there, not just sort of the coordinate shape data. Age, I think, is a little more tricky because I think even sort of aging the fossils is already a tricky process. And so I'm not sure that I want to necessarily say that at this stage the model can do that pretty well, of sort of introducing age and seeing how it works. We made sure to only use adults here because we were essentially testing this against mature individuals. But it'd be interesting to see how, for example, if we introduce juveniles, and if that can sort of make better predictions for specific individuals in the fossil record. So essentially, I want to test everything, is what I'm rambling on about here. Like, I want to sort of see what parameters do we throw into this model that will make it even sort of more efficient, and how do we sort of highlight which parameters, which predictors are better for different hominin species.
Thanks. Yeah, it's an amazing sample that you've got to work with, that, you know, the recent humans that you have so much information about. I won't hog all the questions. Peter, you've got a question.
Thank you. So the 3 reconstructions you showed us early on between yourself and your colleague look like fairly subtle differences. And it looks like your model also comes up with fairly subtle differences. I'm wondering how all of these errors and differences compare to just within species variation, right?
Right. So this image, for example, so it is somewhat fairly subtle differences, but not entirely. So there were, in the sort of paper that we published, there were actually some pretty distinct differences between the 2 reconstructions that were created. They're not pretty visible here, but essentially the shape of the inlet and the outlet of the 2 reconstructions actually like kind of lead to slightly different interpretations about Neanderthal pelvic anatomy, particularly the outlet. The inlet was pretty similar, but the outlet was, so for example, the reconstruction 2 outlet was essentially had the same sort of anterior posterior medial lateral sort of ratio as the Yorak one, but the first reconstruction actually creates a rounder outlet, so a more anterior posteriorly elongated outlet. And you know, subtle as that might seem, it could really sort of, if we're thinking about the hypothetical female, it could really lead to different interpretations about like childbirth mechanism. So how is the infant rotating within the pelvic canal? So because of that, while subtle, some of those differences have sort of bigger biological implications. But when we're talking about like the error and the comparison, essentially, again, it's hard to say for, you know, fossil hominins from which you have very little sort of aggregated information about any one species when it comes to sort of these sort of fine detailed biological parameters. But I essentially think that the sort of performance of the benefit of the reconstruction method, so the benefit of the predictive model, is to essentially give you sort of another line of evidence about where you're placing particular bones and particular elements. So it's not essentially saying just use the sort of predictive model, but it sort of gives you a sort of a narrows your parameters. With Kabara, you're pretty fine. You have that sacrum there. It's pretty complete. And so you know it kind of, you have a good idea of where it fits. With the model, what we're trying to sort of replicate is a situation where you don't have that sacrum. And I would imagine that if you didn't, it'd be a much, much broader sort of range of variation that both of us might have come out with. We use Kabara because we know the Kabara has it there. But other sort of fossil reconstructions have much broader uncertainty and error. So I was looking at, I'm trying to remember which individual it was. That was, I think it's Afarensis, so the Lucy fossil pelvic reconstructions. There's quite a bit of variation amongst the different reconstructions. It's partially due to, you know, the lack of missing, the sort of the lack of certain elements. And so Kabara is lucky in that, like, it looks like, well, it's just like light error, but in other fossils, it would actually do if we could get the model to work for other fossils, I think we would actually benefit a lot from hinging essentially the reconstructed size better, because there's much more variation for other fossils. So there's a much, much higher level of uncertainty at those levels. I think I see another hand. I'm not sure.
Thanks. Tal, do you have a question?
Yes, can you hear me?
Yeah, absolutely.
Oh, thank you. I just have a question. So I understand this is a regression formula or similar to it. Once this formula has been refined to a good, very kind of accurate standard, do you think this could tell us a lot about behaviorism in Neanderthals? So kind of extrapolating off the position of the pelvis, could it give us an idea as to how they walked, how they moved, and from there could that extrapolate further into behaviorism?
Oh, that's the end goal at the end, right? Like that's actually what we're trying to get at. It's not sort of predictive modeling for my benefit. Trust me, I would not do this for my benefit. But essentially, yes. So what we want to get at is if we can make more accurate reconstructions, or at least more confident reconstructions, then essentially we have a better way. We're better situated to understand the biological processes that the pelvis influences. So, like I mentioned before, right? The difference between these two reconstructions and they're sort of the outlet dimensions has implications for childbirth rotation, sort of rotational process in childbirth for those individuals, which means, right, you could actually start to say something about, was childbirth more difficult? Did you need assistance or other things? The shape of the pelvis as well, the way the sort of bones tilt, essentially could give you a lot of information about locomotion habits. What was their gait like? What were the mechanisms of their walking? How much sort of energy were they expending when they're walking? And these are all questions that many people are trying to answer. I'm just sort of trying to be, sort of throw my little contribution into the ring and say, if we have better reconstructions, maybe you can actually answer these questions better. If we, and especially because, again, Kabara is a lucky individual, right? We have most of the bones there. But for many individuals and for many species, we just have really sort of random fragments, right? One ilium here, maybe a little ischium over there. And the idea is, if we can fill in those ones more, then we have a greater sample size to work with. And we're not sort of left working with like singular individuals, right? There's so many other Neanderthal pelvises that I would like to apply this to then give us like a bigger reconstruction sample size that we can then sort of make better statements about biology.
We got any more questions for Mayowa? I was just wondering if you've got any other fossils that you're working on at the moment. Have you managed to get to Bun or anything that you could try this out on?
So interestingly enough, this conversation actually came up very recently for me. So here at Duke, got to sort of have a conversation with Steve Churchill, and we were thinking about actually testing it out in some of sort of the fossil specimens in his lab to see how they sort of work. So I'm actually kind of very interested in looking at some Australopithecus samples and see whether, I really want to see how well it works for slightly more different morphology. I want to see how well it does or how well it doesn't work. To be fair, I didn't expect it to work well, even for the Neanderthals. Maybe I'm just pessimistic, but I didn't expect it to work that well. And so I actually want to see what that era would look like for different hominins. Yeah, absolutely. If I can find time from my other pursuits.
Yeah, maybe you can tell us about what you're doing in a minute, like future work at Duke, but Peter also has another question.
Absolutely.
Thank you. So it looks like your model tries to find essentially the plane of symmetry and it's using the whole side of the pelvis. Would you get a more robust model if you were just using, if you kind of ignore everything that isn't a contact?
If you ignore everything that isn't a contact?
Yeah, so you only look at the contacts to essentially place these things and find the line of symmetry through the two sides of the pelvis.
So essentially, yes. So it's the line of symmetry between the two sides, of course, removing the sacrum and everything. Part of doing that was part of why I took some of that data on the pubic symphysis, because that allows us to sort of have one of those articular points, because essentially, I mean, it's just a rotation, right? So you have one of those articular points, you could essentially just figure out in which plane does that rotate, and that carries the rest of the pelvis along. So yeah, I think I don't know necessarily that it makes it a better prediction. I think it is necessary and to have some of that information there, but I don't know that removing the other sort of landmark data particularly makes for a more robust model. I think it's necessary to have the articular surfaces, or at least one or a couple of them. I don't know that it is necessary to remove the other sort of data, especially because, again, I'm only sort of using the right side. So it's just one bone. that is being sort of thrown into this model.
Presumably any natural asymmetries would occur away from the joints though, so your model might be more robust if you're not including things like the iliac blades, which, you know, may be asymmetric. Yeah. Does that make
Okay,
sense?
I see what you're saying. Oh yeah, I definitely see what you're saying. That could essentially probably do that's actually a good point, that could essentially create a better line of symmetry. At the end of the day, though, it would still be important to essentially reflect the entire bone to the other side. So because this is a bilaterally symmetric reconstruction process, we're assuming the other side doesn't exist. So regardless of whether or not it's going to be off, there's nothing to offset it to in a real fossil. It does that maybe in our model, and that'll be something to test out, definitely. I'm actually interested to see if I only use the articular joints, whether or not it actually creates a, sort of lower prediction error. That'll be interesting. Yeah, I'm actually happy to test that out. Actually, I'm working through it now, and I'm like, there's something interesting to test out.
Thanks.
Are there any more questions from anybody? If you wouldn't mind then, Mayowa, maybe you could just tell us what you're working on in your new postdoc and how it relates to what you've done before.
So interestingly, I'm still working in the pelvic space because I just think that it is the most fascinating region of the human body. I've staked my claim that it is what it is. The rest of you work on other things, figure that out. But I have sort of switched gears a little bit. So I'm right now at Duke. I'm in the animal locomotion lab, so the Daniel Schmidt Animal Locomotion Lab. And what we do there is a lot of biomechanical analyses, so gait analysis. And so what I'm actually working on right now is a project in collaboration with the Duke Health, so the hospital at Duke. I'm working with surgical oncologists at Duke, as well as essentially prosthesis surgical implant companies. So we're working with Restore 3D, which is the surgical implant creation company. And what we're trying to do is, so we're trying to help them improve surgical implants as well as improve actual surgical procedure for patients that have had or that require a hemipelvectomy. So these are individuals who have a tumor or some cancer in their hip bone that requires for that entire hip bone or parts of it to be resected. And essentially what they do is they have these 3D printed metal implants, the most expensive thing, block of titanium we've ever seen, that essentially is fit sort of attached to the pelvis, sort of reconstructed. And what they've been having a problem, and this is a problem that has existed since this procedure has existed, is that whenever they reattach the abductors, so the hip muscles, after a while they fail. Without doubt, the muscle attachments will eventually fail. And so the question that they came to us with was, do we have to reconstruct this individual back to its anatomy, to the sort of anatomy that we know that this particular individual had, or are there other configurations that we could sort of work with that would maybe reduce the occurrence of muscle failure? And are there other designs for these prostheses that might actually be more biomechanically efficient for these individuals? And of course, they came to the evolutionary anthropologists because if there's one thing that we know is pelvic variation. At least that's what I've been digging into for the past couple of years. And so what I'm trying to now do is with my team is to essentially pull from the fossil record and pull from everything that we know about human pelvic variation and to try to pull elements that might actually be more biomechanically sort of efficient for this individual. Because when you essentially introduce a new surgical implant, no matter how perfect it looks like the bone that you took out, there's going to be biomechanical deficiency. So what we want to do is, are there things that we can sort of pull from the fossil record that can sort of reduce that biomechanical deficiency? Is there something about Australopith's pelvic shape and muscle attachment that might actually maybe work better for an individual who now has a reconstruction that maybe tilts a little bit more flared, like an Australopith, for example? And so we're sort of trying to pull from that fossil record to do some gait analyses to sort of help improve both the actual surgical procedure in the room as well as the creation of the implants by the company that does it. So slightly big, I'm not going to call it a slightly big pivot. It's a big pivot, but it's sort of like an interesting way to apply a lot of sort of what I've been thinking about for the last couple of years, and to sort of find a way to apply it in a clinical setting to sort of actually, you know, help patients have better quality of life.
That sounds fascinating, and it's a really rare application for evolutionary anthropology, so that's very cool.
Yes. Yes, it was very, very different from what I thought I would be doing, and I think that's why I wanted to do it. It was I've never sort of thought about applying evolutionary anthropology in that sort of way, and I was really interested in the opportunity to do so. I am still also going to be working on this project, extending this into the future, probably working with Steve Churchill on that in the anthropology department as well, to sort of, you know, refine this method. So I'm still very, very much interested in this aspect as well. Just, you know, science.
Yeah, in all your free time, you'll be fine.
Yeah, I'm sure I'll find some time to do this.
Any final questions? Okay, well, I'll just say thank you very much, Maya. That was a fantastic talk, and thank you everyone for joining us. I'm going to turn off this recording.